Hello, everyone. My name is Sarah Hermann, and I'm an assistant professor in ecology and entomology here at PSU, and the head of the arthropod ecology and trophic interactions lab that you will be touring today virtually. I'm broadly interested in understanding how population and community dynamics are shaped by species interactions. And in the lab, we study these interactions through the lens of pest organisms and their predators. Our research addresses how top-down forces influence insect ecology, contribute to trophic cascades, and structure ecological communities. In studying these interactions, we use an integrated approach, combining manipulative laboratory experiments with field studies to identify and quantify the way in which insects interact with, respond to, and are changed by their environment in both agricultural systems and natural systems. Current and ongoing work in the lab focuses on how insect predator-prey ecology and the underappreciated role that the risk of predation has on insect behavior, physiology, and ultimately fitness. I'm also the instructor of two courses here, Integrated Pest Management and the Applied Biostatistics and Reproducible Research in R class. And I also serve as the co-founder and head of the Diversity, Equity, and Inclusion Committee in the Ecology program. Nice to meet you.
Hi, my name is Des Kansman, and I am a postdoc in the Hermann lab. I just started back in fall of 2020 after finishing a PhD in insect ecology and abiotic stress interactions with Debbie Finke. I chose the Hermann lab because Sarah's research is really dynamic and interesting, and she is also a fantastic individual to get to work for. So my work right now is focused on chemical ecology and how chemical ecology mediates the non-consumptive interactions between predators and their prey.
Hello, I am Jorge. I'm a new PhD student at Dr. Hermann's lab. As an agricultural engineer and entomologist, I am interested in applying chemical ecology into integrated pest management practices for more sustainable agricultural systems. I choose this lab because I find here the opportunity to grow and learn in a multidisciplinary environment.
Hi everyone, my name is Lily Germeroth. I'm a master's student in the ecology program, and I just began here last fall. And I'm in Sarah's lab looking at predator-prey interactions in the monarch milkweed system, and I'm having a really great time. And I chose Sarah's lab because she is a really positive, supportive mentor, and the research that she does is really dynamic and interesting and has a really strong ecological core to it. So this is a tool that we use to look at anti-predator behavioral and physiological changes in aphids. So there's aphids there on that leaf, and there's a layer of mesh in between them and their lady beetle predator. This way the aphids can smell the predator but not be eaten by it. This is another example of an arena to look at anti-predator behaviors and to isolate non-consumptive effects. So you can see ladybug predators on top, and there's aphids on those leaves in the bottom. This is another tool that we use in lab, and it's called a Y-tube, and we use it to assess insect choice. So here we're looking at one arm pumping in smells from a host plant that is infested with natural enemies, and the other has plant scents that are free of natural enemies. And that black dot is an aphid, and we can see that it chooses one arm over the other.
The Hermann lab also has an electrical penetration graph, or EPG, which we can use to measure aphid feeding behavior. You do this by creating a circuit between the plant and the aphid using a probe that I joke is an aphid fishing lure, and attaching an aphid to the end of that fishing lure, which is gold wire with silver glue. The aphid is then placed on the plant, and the probe is placed within the electrode. And with a probe that is placed in the soil, you now have a full circuit in the plant. And that's how we EPG.